is the history of Nipah virus. We might be wondering, what is the Nipah viral disease? When and where was Nipah virus discovered? Is there any other region that may be affected? And how was it transmitted? Here's the answer. Nipah viral disease is an emerging disease that is actually caused by Nipah virus. Its scientific name is Nipah virus. The RNA virus of genus Nipah virus in Paramyxoviridae family was transmitted by a specific type of fruit bats, and mostly it is Theropus species. The first discovered was in 1998. It is located in one of the villages in Malaysia, which is Kampung Sungai Nipah, Perak. It was found during an outbreak among pig farmers. In 2001, Nipah virus was recognized in Bangladesh. Also, this disease identified periodically in eastern India. There is evidence that the virus has been found in no natural reservoir. There are other species of bats, including Theropus bat species, in a few countries. This virus is a zoonotic virus which is transmitted from animals to humans. Not just that, it also transmits through contaminated food. It is also transmitted directly between humans. If someone has been infected, be careful because it may affect you with Nipah virus by their body fluid, such as blood, nasal, and urine. You might end up being sick. During the first outbreak in Malaysia, which also affecting Singapore, the infection was resulted by a direct contact with a sick pig. While in Bangladesh and India, they were infected by consuming raw fruits or which had been contaminated with urine or saliva from fruit bats.
Some additional facts.
As you know, Nipah virus infects its host cells via two glycoproteins, which are G and F proteins. The G glycoprotein mediates attachment to host cell surface receptors, while F protein makes fusion of virus cell membranes for cellular entry, which cause people who got infection associated with encephalitis, which swelling of the brain that can cause mild to severe illness and even death.
Next one, we will go through on people who are at high risk to be affected by Nipah virus. The first one is abattoir workers. During 10 to 19 March 1999, 11 workers in one of two Singaporean abattoirs developed Nipah virus associated encephalitis or pneumonia, resulting in one fatality. The second one is pig farmers. The Nipah virus was first discovered in Malaysia in 1999 during an outbreak among pig farmers.
The third one is people who consume palm sap.
A 2004 Bangladesh outbreak was traced back to humans consuming dead palm sap that had been contaminated by infected fruit bats. Moving on to the next part, which is how long does Nipah virus can be detected by people. The incubation period, which is the interval from infection to the onset of symptoms, is believed to range from 4 to 14 days. However, an incubation period as long as 45 days has been reported. The next one is the symptoms when someone gets Nipah virus. Symptoms may initially include one or several of the following, such as sore throat, fever, myalgia, vomiting, cough, and headache. Real-time polymerase chain reaction (RT-PCR) can be conducted to diagnose Nipah virus infection from throat, cerebrospinal fluid, nasal swabs, urine, and blood. After recovery, testing for antibodies is conducted using an enzyme-linked immunosorbent assay (ELISA). Currently, there are no licensed treatments available for Nipah virus infection. Treatment is limited to supportive care, including rest and hydration. The natural host of the Nipah virus is the fruit bats, which is the Theropodidae family.
Now, I'm going to share the impact of the disease to human in terms of health, socioeconomy, and emotion. The first impact disease to human is health. Infection of Nipah virus can cause mild to severe disease, including swelling of the brain, which is encephalitis, and potentially death. In a few cases, infected people have also shown symptoms of myocarditis, which is inflammation of the heart. The second impact to human is socioeconomy. In 1998, an outbreak of Nipah virus occurred in several pig farming communities in Bukit Pelanduk, Negeri Sembilan, Malaysia. The outbreak in Malaysia resulted in 265 cases of acute encephalitis with 105 deaths and near collapse of the billion-dollar pig farming industry. The next impact is emotion. The most reported emotions were loneliness, with total 87.3% and fear, 86.3%. Some of them experienced fear of death during isolation stay, and several patients had lost their family or friends to Nipah virus infection.
The outbreak has been controlled by culling pigs in the states of Perak, Negeri Sembilan and Selangor to prevent the virus from spreading further. The Ministry of Agriculture's Department of Veterinary Services was given the authority to cull all unhealthy and in-contact pigs. Approximately 1.1 million pigs were culled in total to control the outbreak. A national surveillance is currently being developed to eliminate any more affected herds. It was primarily classified into three groups of healthcare sector surveillances, namely disease surveillance, patient surveillance, and high-risk group surveillance. The National Swine Testing and Surveillance Program included a surveillance program for all pig farms, an abattoir surveillance program based on random blood sample, and a high-risk farm testing protocol. When handling sick animals or their tissues, as well as during slaughtering and culling procedures, gloves and other protective should be worn. Close and protected physical contact with people afflicted with the Nipah virus should be avoided. Hand washing should be done after caring for or visiting sick patients on a regular basis. This practice is used to reduce the risk of human-to-human transmission. Care workers who are caring for or handling specimens from patients with a suspected or confirmed infection should follow compassionate infection control procedures at all times. Samples taken from people and animals suspected of being infected with the Nipah virus should be handled by competent personnel in properly equipped laboratories.
Another additional facts.
There are some prevention that we can do to reduce the risk of infection in people. First, reducing the risk of bat-to-human transmission by decreasing bat access to dead palm sap and other fresh food products. Keeping bats away from circulation sites, and always wash and peel fruits before consumption. And also, fruits with a sign of bat bites should be discarded immediately. Second, reducing the risk of animal-to-human transmission by wearing gloves and other protective clothing while handling sick animals, and people should also avoid being in contact with infected pigs. And lastly, reducing the risk of human-to-human transmission by always wash the hand or use hand sanitizer after caring for or visiting sick people.